Hello, NSM. Apa kabar? I'm so happy to be there with you, even if it's through a recording. I'm going to give you a scientific update on metabolic improvement aspects with Palatinose, which is the commercial name for isomaltulose. Dr. T, NSM Council, and organizers of this conference, thank you very much for this fantastic opportunity. And Benio, thank you for your support. Let me start off with making my disclosures. I have received isomaltulose from Benio for the student projects that I supervised while at IMU. I have received no funding from the sponsors for this presentation, and they have not been involved in conducting the review or in collating the results for this presentation. Here's the presentation flow that is planned for today. First of all, let's get to know isomaltulose. Then let's quickly review the evidence in the three areas that are listed here. We will look at future potential areas to explore or approaches that we could take in future. We will also look at the state of translation and food applications of isomaltulose and finish off with take-home messages. First things first, let's understand the purpose behind this rush for sugar alternatives. Sugar, we know, is an indispensable part of our diets. It's impossible to completely take it off of human diets. But excessive sucrose intake and certain populations have postprandial glycemic surges, which lead to non-communicable diseases. Therefore, we are looking for alternatives to sucrose. In this context, it's very interesting to observe that recent guidance, conditional guidance from the WHO, gives us certain uncertainty about the use of non-nutritive sweeteners. It is here that isomaltulose comes as a promising alternative with its suitable sweetness, with its potential health benefits, and also a feasible production cost, which leads to its affordability. And now let me introduce you to isomaltulose, which I call a biochemist delight. And here you see Dr. Peter Barling, a retired professor of biochemistry from IMU, visibly happy when talking about isomaltulose. Why is that so? Because isomaltulose is a really interesting disaccharide. So as a disaccharide, it is composed of two monomers, glucose and fructose, exactly the same monomers that you would find in sucrose. So what's different then? It's the way that they are bonded. So the glucose and fructose monomers are bonded with an alpha 1,2 linkage in sucrose, whereas in isomaltulose, the two monomers are bonded using a 1,6 linkage. It is this different bond that actually gives isomaltulose its differential properties. Now let's learn a little bit about the digestion of isomaltulose in small intestine. Both isomaltulose and sucrose are hydrolyzed by the sucrase isomaltase complex, but like you can see the site of hydrolysis is slightly different. And the hydrolysis of sucrose is 5 times faster than that of palatinose. Therefore, what happens is when somebody consumes isomaltulose, it is hydrolyzed much more slowly in the small intestine compared to sucrose. And this gives isomaltulose its low glycemic properties. So this is the Nutrition Society of Malaysia's conference. So let's look at what isomaltulose means to all of us as nutritionists. While it is completely digested, because of the rate of hydrolysis of isomaltulose, it is a slow-release carbohydrate, and it provides glucose and energy at a slower pace and over a longer period of time, which is typical of a low glycemic food when compared to sucrose. In 2018, I reviewed the health benefits of substituting sucrose with isomaltulose along with Dr. Amudha Ramadas and Dr. Chang Sui Kit. And like you see here, it has been cited for around 47 times since then. But 5 years have passed, and we know a lot of things can happen in 5 years, especially in terms of life sciences. So therefore, it's time for a rapid update, and which is what I aim to do in this particular presentation. So let me tell you a little bit about the process of conducting this scoping review. I used the PubMed database. I searched for studies which had isomaltulose or palatinose occurring anywhere in the paper. I did not use any health outcomes because I did not want to restrict the papers, but I wanted to widely scope the papers to see the evidence. Of course, I looked for papers which were published after 2018 because this is a scientific update after 2018. And I was only looking for studies from providing human evidence. So after doing all this, there were 43 studies. And out of these 43 studies, I had to exclude 26 studies because of the reasons that have been listed here. And we have 17 studies that are included in this scientific update. So you will see much of the evidence comes for postprandial glycemia or postprandial insulinemia. There's sufficient evidence to look at how isomaltulose performs in terms of body weight management or other related variables such as appetite or satiety. And there is also some interesting evidence in terms of novel cardiovascular risk, which I shall show you shortly. So because I used only one database, I wanted to make sure that it was wide enough. So I also did a bibliometric analysis using Research Rabbit. And you will see the green ones were the papers that I had in my database, and the blue ones were the papers I did not have in my database. And some of them were older studies or were definitely not looking at the categories of variables that I was looking at. So instead of the 38 studies that Research Rabbit pulled out, I already had 43 studies. So I was pretty happy to proceed with reviewing the evidence. First, let's focus on isomaltulose and body weight management. Okay, now let me collate the evidence for isomaltulose and body weight management here. I use a similar template for all the health outcomes that we are evaluating today. So I would like to take a few minutes to explain this table to you. So we have the reference, the country, and the study design. Under the study design, if it's a RCT, which provides the highest level of causality, I have used bold font to bring it to your notice. And also when the participants included patients with disease, I have highlighted it in bold font once again for your convenience. I also list the control against which isomaltulose was compared in each of these studies. And here we have the variables that were studied under a particular health outcome. So let me tell you how I color code the evidence for these variables. So when you see a cell in green, it means isomaltulose has performed better than control. When you see a cell in gray, it means both the control and the isomaltulose had similar effects. If I had color-coded a cell in red, it means the control did better than isomaltulose, which is not the case in any of the studies that we see here. And so when you see a cell which is white in color, it means the particular variable has not been studied in that particular study. And this makes sense because in acute studies, there is no point studying body weight because body weight doesn't change within a 2-hour period of time. What we see here is both RCTs showed beneficial effects of isomaltulose in terms of appetite. Isomaltulose, when compared to sucrose or maltose, was useful in reducing appetite and therefore energy intake. And the longest study that we have shows that fat oxidation was increased by isomaltulose in comparison to sucrose and led to a decrease in body weight. For each of the health outcomes, I have selected a few studies where we could learn a little bit about the study design and how the findings were interpreted. So here is the first study by Lightoller et al. In 2019. It was a 12-week study, and I also summarize the study design using the PICO format. P for the population, I for the intervention that was used, C for the control, and O for the outcomes, main outcomes that were studied. So in this particular study, 64 healthy adults with overweight and obesity were put on a 12-week RCT. It was a double-blind RCT where people were either given 40 grams of isomaltulose per day or 40 grams of sugar. And at the end of the 12 weeks, the researchers looked at weight loss. So here you have the results in blue for isomaltulose and the results in red for sucrose. And what the authors concluded was the isomaltulose may be more effective in promoting weight loss in comparison to sucrose. So here's the other RCT from the 6 studies we looked at for isomaltulose and body weight management. This is a 10-week study which was conducted in Spain. It's a very interesting study. The population included 41 type 2 diabetes patients with overweight or obesity. And the intervention was alcohol-free beer, which contained modified carbohydrates, isomaltulose and maltodextrin. The control was similar alcohol-free beer, but it did not have the modified carbohydrates, so it had maltose. So the authors compared these two sugars in terms of biochemical markers, anthropometry, energy intake, and satiety. So this was a crossover trial with a washout period of 6 to 8 weeks. So people took one of these for a period of 10 weeks. So what happened was when they were on the IM part of the trial, they had significantly reduced appetite and they reduced their energy intake. And specifically, they found that the energy intake coming from saturated fatty acid was reduced. And isomaltulose also significantly reduced insulin resistance during that period of the trial. So the authors conclude that isomaltulose may be more effective in weight management in individuals with type 2 diabetes. Now let's move on to looking at isomaltulose and postprandial glycemic management. So here is the table that collates the evidence in the area of isomaltulose and postprandial glycemia. You see there's a lot of studies here, and then you have only one trial, which is an RCT, and this was the same Spanish study that I showed you earlier. So we are not going to discuss this anymore. You will see there is a lot of studies in healthy people, but there are few studies in people with non-alcoholic fatty liver disease, in those with prediabetes, diabetes, mildly hypertensive adults, and those with type 2 diabetes again. So when you look at the color coding, you can see that the evidence for postprandial glycemia, reduced postprandial glycemia with isomaltulose is pretty consistent, and similarly with insulin. So a few studies also looked at gut hormones, which included GIP, which is the gastric inhibitory polypeptide, and GLP, which is the glucagon-like peptide. These are very expensive hormones to study, and very few studies have used these markers. So GIP is an inflammatory marker. GLP-1 is more is an anti-inflammatory marker, if I could make that simple for you. So having lower levels of GIP is good, and having higher levels of GLP-1 is good. So if you look at it, the evidence for isomaltulose to beneficially modulate GIP to GLP-1 ratio is also present in these studies. Now let's look at this very interesting study that comes from Professor Henry's research group in Singapore. What the authors tried to do was look at whether isomaltulose and beta-glucans could be used as novel food ingredients as part of a low GI diet. So what they were trying to prove here is whether using these ingredients they could mimic a low GI diet. A low glycemic load diet keeps your glucose stable throughout the day. It reduces glycemic variability in comparison to a high GL diet. So here is what they did. They did a randomized single-blind control crossover study in 12 healthy Chinese men. So they have this menu that was provided to the subject. So you could look at the differences. You had one for the low GL ingredients, and you had one whole list for the high GL ingredients. For the breakfast, both groups received yellow noodles. When I say both groups, I mean in both periods of time, people received yellow noodles, but the LGL ingredients contained beta glucan. At lunch, the low GL group received parboiled basmati rice. The other group received glutinous rice, which we know has high GI. Apart from that, both groups received chamomile tea, whereas the low GL group received isomaltulose to sweeten their tea. The other group received sucrose. Snack once again had isomaltulose as a sweetener versus sucrose. And finally, in the dinner, while the low GL phase contained pad thai glass noodles served with tofu, the other phase contained teriyaki chicken with rice. And once again, there was this chamomile tea with the isomaltulose or sucrose. So here are the results for you. So you will see that the results that you see here are pretty close to the theory picture that I showed you here. So you have the low GL diet that was done with isomaltulose and beta glucan here, and the ones with the bold line without the breaks is the high GL diet with the glucose. So what they found was there was the lower maximum level of glucose over the day, and it was highly significant. And this was especially significant after dinner. Probably the effect keeps adding on to the next meal. And more importantly, they looked at MARGE. MARGE gives you the glycemic excursion, the maximum amplitude of glycemic excursion. You do not want to have a large swing throughout the day. And they found that there was balanced level of blood glucose when people were on the low GL diet that included isomaltulose and beta glucan. I want to bring to your attention this excellent systematic review and meta-analysis that was published by Z et al. In 2022. So this is quite recent here. This meta-analysis included 11 RCTs, which included 175 participants in total, all of whom were adults. And the participants came from 4 different countries: Japan, Brazil, Germany, and the Netherlands. So the participants were healthy or could have had type 2 diabetes, impaired glucose tolerance, or hypertension. So the authors collated the results in terms of 3 specific outcomes. The first one is about the attenuation of glycemic response in the immediate 30-minute postprandial period, where they compared isomaltulose with sucrose. And they found that isomaltulose definitely reduced the glycemic response in comparison to sucrose. And they have graded the level of evidence to be moderate. The second aspect that they looked into was whether the glycemic response was prolonged over a longer period of time, and also looked at the insulinemic response in comparison to sucrose and found it attenuated. For these 2 outcomes, they find the level of evidence to be low. And additionally, they also identified populations in which this substitution would be especially beneficial. This slide is also taken off the same systematic review, where Z et al. Very clearly visualize specific population groups in which a substitution of sucrose with isomaltulose not only decreases the glycemic response, they also have a sustained glycemic response and an attenuated insulinemic response. And this population which specifically benefits from this substitution are patients with type 2 diabetes, impaired glucose tolerance or hypertension, older adults, adults with overweight and obesity, and particularly Asians. So for attendees of this conference, this may be very, very interesting because Asians have inherent insulin resistance, and that is why the results in the Asian populations are sometimes more higher in magnitude compared to those found in Caucasians. And also, very importantly, for those who are interested in this area, more RCTs using standard mixed meals containing isomaltulose. So they are looking at more guidance in terms of how to translate this evidence into daily use. Trust in science is built when we can mechanistically explain why something happens. So in the next few slides, I'm going to provide you some evidence of how and why isomaltulose behaves in the way it does. I'm going to start building the mechanistic evidence with this small study that came from Brazil in type 2 diabetes patients. So this was a randomized double-blind crossover study where the subjects were in one phase fed a diabetic specific formula with isomaltulose, and in the other one another commercially available standard formula. If you go and read this reference, you are able to identify what those formula were. Because the ONS had a lower GI value, you would expect a lower insulin response. But here is where I want you all to pay attention, because this is a very interesting mechanistic point of view. Here we are going to talk about GIP, which is the gastric inhibitory polypeptide, which we touched upon in one of the fewer slides. So like I told you, GIP has inflammatory properties and also increases appetite. So in this particular experiment, the authors found that the diabetic specific formula could also lower the GIP response postprandially. And they were able to correlate this with lower subjective appetite scores from the subjects as well. So this means that diabetic specific formula, which contained isomaltulose, apart from lowering their blood glucose response and insulinemic response, was also lowering the endogenous secretion of GIP and therefore reducing the appetite, and potentially this could lead to lower energy intake. In this super busy slide here, which I wish I had done a little bit better, I am trying to tell you the story of incretins and how they modulate the effect of carbohydrates based on their glycemic index. When somebody consumes high glycemic carbohydrates, we know that it creates postprandial hyperglycemia over the period of time. But nutrient intake can also trigger the increase in the secretion of GIP when somebody consumes high GI carbohydrates, and GIP is secreted in the intestine, small intestine, in the K cells, in the proximal parts of the small intestine. Increased secretion of GIP leads to decreased lipolysis, increased inflammation in the postprandial period, and increased insulin secretion, which leads to decreased fat oxidation and also accumulation of fat in the liver. In the brain, this can lead to increased appetite and increased food intake. So, as a cumulative effect of all of this, postprandial glycemia and an increased GIP to GLP ratio can lead to body weight gain and its consequent problems, including diabetes and cardiovascular diseases. So, even before the onset of cardiovascular diseases, as an interim marker, you are able to see what happens when somebody consumes chronically a high GI diet in terms of the endothelial dysfunction. I will show you some examples of this in the later slides. So the basis for many of this would be the postprandial oxidative stress and inflammation which accumulates over the period of time. However, when somebody consumes lower GI carbohydrates, more GLP is secreted. GLP is also secreted in the small intestine, but it is secreted in the distal part of the small intestine by the L cells. So what GLP does is it reverses many of the functions of GIP. So a lower GIP to GLP ratio has cardiometabolic effects. After 2016, there has been an interest to understand how low GI diets work. So one of the most interesting hypotheses that has been put behind is the incretins. So there is an increased interest in studying GIP and GLP. So in these studies that you see here, much of the hypothesis is based on studies that compare isomaltulose with either a sucrose solution or another standard formula. So all the effects that we have seen that have been demonstrated in many of the studies which document the mechanistic action of low GI diets on GIP and GLP have used isomaltulose. The reason that most of these studies used IM as the low GI alternative or ingredient is very easy to understand, because IM facilitates creating a low GI alternative to a standard formula or a high GI formula without changing the other components. And from all the evidence that we see here, we can read that isomaltulose may lower GIP to GLP-1 ratio in comparison to higher GI alternatives, and this can have cardiometabolic advantages above and beyond the management of postprandial glycemia. The third and final area of evidence we are going to look at is isomaltulose and novel cardiovascular outcomes. Three of the studies that were included in this review looked at some novel outcomes. Two of these studies looked at endothelial dysfunction using different variables, and one of them looked at bile and fatty acid metabolism. So this is a small study, but it is spectacular nevertheless, because it was conducted only in 5 male subjects with non-alcoholic fatty liver disease. So the researchers fed the participants either 20 grams of isomaltulose or sucrose, and in the postprandial period from baseline to 15 minutes, they withdrew plasma and created a metabolomic analysis where they studied all the protein markers. So here what they did is they looked at the number of altered metabolites in each of the pathways that you can think about. So I'm labeling the first two to you. The first one is the bile acid pathway, and the second one is the fatty acid metabolism, the metabolites which were involved in this pathway. So they also studied changes in blood glucose, insulin resistance, and specifically studied serum C-peptide immunoreactivity, which is abbreviated as CPR. Interestingly, no difference in blood glucose was found when people were fed IM or sucrose. However, CPR level was significantly reduced, which shows that there was a decrease in insulin resistance in these patients. And there was 52 metabolites. There were 52 metabolites that had significant alterations, and they were mostly linked to bile acid and fatty acid metabolisms. So what the authors concluded was that isomaltulose improved insulin resistance in non-alcoholic fatty liver disease patients, and this was done through altering bile and fatty acid metabolisms. The next two slides, we are going to study a concept called flow-mediated dilatation, that is abbreviated as FMD. So what does FMD measure? FMD measures the ability of your vascular endothelium to release nitric oxide. And why is this important is because this release of nitric oxide gives the ability of your endothelium to support a transient flow stimulus. So what do I mean by this? Imagine I place a blood pressure cuff on your forearm. I pressurize it and I stop your blood flow for a few minutes. Then I suddenly release it. Then what happens is your blood should flow out a little bit more faster than it usually does. And you should have sufficient nitric oxide released by the endothelial cells in your blood vessels in order for it to dilate sufficiently to accommodate this sudden increase in blood flow. That is FMD measurements for you. So FMD measures vascular health, and it is an early marker for cardiovascular disease. Also, by measuring the FMD, you can see if a intervention is reversing some of the vascular disease. So it's a very early marker. This first study by De Groot et al. Compared isomaltulose versus sucrose on endothelial function in overweight adults. The study was an acute RCT and had a fairly decent sample size. In the study, they looked at 50 grams of isomaltulose versus 50 grams of sucrose, and they studied endothelial dysfunction postprandially. If you remember my slides on incretins, I told you about how in the postprandial period, the oxidative stress and inflammation can actually decrease flow-mediated dilatation. The endothelial function is reduced. So what the authors here wanted to see was whether IM improved FMD postprandially in comparison to sucrose. So what they found was IM reduces the decline in postprandial endothelial function in comparison to sucrose. And they also noted this benefit was specifically seen in individuals with impaired glucose tolerance. Therefore, in people with impaired glucose tolerance, which can be obese or overweight adults or those with prediabetes, and definitely the diabetic population as well, isomaltulose may also promote cardiovascular health. So this study also kind of supports the meta-analysis that we alluded to earlier. This study by Kobayashi looked at a very related concept, which is called arterial stiffness in 10 healthy, normotensive, middle-aged and older adults. So how do you measure arterial stiffness in people? We use something that's called pulse wave velocity. What is pulse wave velocity? It's the speed at which the blood pressure propagates through the circulatory system. Very high levels of pulse wave velocity indicate somebody is at high risk for cardiovascular diseases. Not only that, damage to organs and also all-cause mortality. So in the postprandial period, what you can see here is, in comparison to sucrose, isomaltulose is able to reduce the pulse wave velocity. And this was significant. And this was also correlated with decreased blood pressure, systolic blood pressure. So the authors went on to conclude that isomaltulose may have significant clinical or dietary implications for middle-aged and elderly patients who are normotensive, probably. After we have reviewed the evidence that we've seen today, we are able to add to Ahmed et al.'s synthesis of evidence from human trials on isomaltulose, which was published in 2022. So here we can look at the evidence in terms of the type of population, the healthy, overweight or obese, and the type 2 diabetes or pre-diabetic population. And we can also look at it in the time frame from the acute standpoint and from the more longer time point. So in healthy people, acutely isomaltulose is able to reduce postprandial glycemia, improve flow-mediated dilatation, and in the longer term, there is also evidence to show that it can reduce insulin resistance. In overweight people, once again, there is evidence for short-term reduction in postprandial glycemia, improvement in FMD, and over the long period of time, there is evidence to show that there is reduced body weight. So here we are talking about isocaloric diets where a higher GI sweetener such as sucrose is replaced by isomaltulose. In type 2 diabetic patients, acutely once again, the postprandial glycemic response is reduced. Interestingly, in the longer period of time, insulin sensitivity improves and insulin resistance decreases in these patients. So with the current evidence, we are able to collate the metabolic actions of isomaltulose in humans. And this is a beautiful pictorial from Vandlar et al., who has shown the mechanistic action of many of the alternative sugars here. And to make it easy for you, I have used these pins to indicate where isomaltulose has significant evidence in the area. So in terms of slower absorption, there is definitely some evidence. There is, in terms of reduced glycemic response, a lot of evidence. In terms of improved fat oxidation and blood lipid improvements, there is evidence for isomaltulose again. And in terms of beta cell protection, there is evidence for isomaltulose. And more recently, isomaltulose has also shown to improve GLP-1 secretion. However, what's interesting is there is a scope of a lot of other variables that we can look into specifically on the Asian populations. Now let's move on into futuristic innovations. Here we are going to talk about how to move nutrition science from the current state of art to something more cutting edge. So we are going to look at some research approaches that will move us into the future. We will also look at how isomaltulose can be used in future. So firstly, I'm going to talk to you about this very innovative approach, which is called the citizen science. So what is citizen science is when the citizens work with the scientists to create the evidence. And this is one step towards creating a large amount of evidence, which becomes very easily translatable. So this is catching up a lot in Europe, and I'm hoping that we will be able to do a lot more of it as well. So in this citizen science project, we are looking at people who are free living, which means what is happening in real life. This novel approach involving citizens has become more possible with technological advances that we have seen, especially the wearable technology. So one of those things that are very useful in today's research is the CGMS. CGMS stands for Continuous Glucose Monitoring System. Some of these small sensors, which are just the size of a coin, can be applied to your forearm. It doesn't even show out of the clothes if you're wearing a sleeve dress. So what it does, it captures your every minute blood glucose data for the 14-day period that it can last up to. And then it can be downloaded onto their computers. So this whole study was a 14-day study, and the CGMS was applied on the first day. And the first day was just about calibrating these sensors. This is very important for data validation. So apart from this, they also use digital food diaries to capture the food intake, and also they were using digitized body weight measurements. So let's see what they did to these 117 adults who took part in the study. On days 2, 7, and 12, they were given a test drink A, which contained 30 grams of Palatinose, which is isomaltulose, both at breakfast and at lunch. And on days 3, 8, and 13, subjects were given test drink B, which contained 30 grams of sucrose. Remember, these subjects are wearing the CGMS. So the scientists can see what happens when they are consuming test drink A or test drink B. From day 4 onwards, the participants were also given 10 grams of Oraf-T, which contains inulin, which is a prebiotic. And this was provided to them at the dinner. So for a 11-day period, they were consuming Oraf-T as well. So at the beginning of the study and at the end of the study, stool samples were collected. Gut microbiota was evaluated using 16S RNA sequencing. Throughout the 14-day period, food intake was also captured using digital technologies. So let's get to the climax of the story after all the elaborate arrangements that the scientists had done. So as expected, isomaltulose, which was fed as Palatinose, resulted in lower blood glucose compared to sucrose. But this was significantly so on days 7 versus 8 and on days 12 versus 13, which means potentially there is some kind of a cumulative effect that happens. And this happened both in breakfast and after lunch. And after people started taking the Oraf-T Synergy inulin supplement, there was an increase in bifidobacteria. Bifidobacteria is a commensal, so it improves health. So these were the two findings from the study. So let me let you in on a secret. The most juiciest part of the study is never provided in the abstract. So never just skim the abstract. The real good pieces of research are towards the end of the results written in very minute print. So here's what we found in this study. So you may ask me, the lower postprandial blood glucose response or having a sustained blood glucose response throughout the day, having less of the glycemic variability, etc., were all studies, were all findings that we saw in the earlier slide. So what's new here? What's new here is the citizen science approach because it showed you the theoretical concepts that have been proved in the lab are also applicable to the real world. And that's an important takeaway. Number 2, consumption of 10 grams of Oraf-T Synergy, which is an inulin supplement, had synergistic effects. And how did it do this? Was by increasing bifidobacteria in the gut. So these are very interesting ways in which we can combine functional food ingredients in order to benefit health. Here is the super interesting part of the study. Among the 117 people who took part in the study, 72% of them were classified as good responders. Who is a good responder? A good responder is the one who responds very well to isomaltulose and has a lower PPG, postprandial glycemia, in comparison to when fed sucrose. So how do we identify those who would specifically benefit from isomaltulose? These were people who had a differential gut microbiota. They had higher firmicutes to bacteroidetes ratio. So what this is telling us is the scope for personalizing nutrition and the scope for using functional food ingredients in personalizing nutrition recommendations. In this final section of the presentation, we are going to look at the real-world food applications of isomaltulose. Before we go into seeing how isomaltulose can be applied in novel food applications, let's revise a little bit about the physiological properties of nutritive sweeteners, a class under which isomaltulose falls. It is a nutritive sweetener because it contains calories. Isomaltulose provides 4 kilocalories per gram, the same that you would find in sucrose and most other disaccharides. However, isomaltulose has a GI of 32, whereas sucrose has a GI of 68, which means the postprandial glycemic response to isomaltulose, as we saw earlier, is only half of that of sucrose. Therefore, the insulin response to isomaltulose is also lower than that of sucrose. And there's evidence to show that isomaltulose can be successfully used in patients with diabetes. But from the food science perspective, it is also important for us to understand that isomaltulose has only half the sweetness of sucrose. Therefore, we should be very mindful of where we can substitute this beneficially. So now let's look at the patent applications for isomaltulose that's available in the public domain. The oldest one that I could identify was filed in 1995. So isomaltulose has been around for a long time and it has been patented for application as food additives, for quality improvement, as nutritional supplement or functional food, or as a flavor enhancer. And the amount of patents that you see is really quite impressive. The FDA approved isomaltulose for use in 2006 and it is generally regarded as safe, GRAS. In 2017, FDA once again approved the use of dried IM syrup and provided it a GRAS number as well. But here's something we must remember, friends, it's a nutritive sweetener. So it is approved for use as a sweetener in solid foods and beverages. But remember, once again, we want to use it at the same level that is recommended for use as sucrose. When it comes to food choice or translating the benefits of novel food ingredients, two things are very important. Number one is the cost. Number two is the taste. So when something is too expensive or not tasty enough, it's not going to get translated. So let's look at these factors in relation to isomaltulose. Number one, isomaltulose is produced via a non-GMO enzymatic rearrangement of sugars. So they have brought the cost under a lot of control. And what literature says is if there is more demand for isomaltulose, this economic barrier can still be slashed. So at this point, isomaltulose costs around 3 times that of sucrose, which is pretty reasonable for a novel food ingredient. But what the literature says is with more demand for it, it could go down more. The second most important thing is the taste. So here is an example of how well isomaltulose does. 8 different sweeteners were compared to sucrose in 3 food matrices, in black tea, in chocolate milk, and natural yogurt. So this is the kind of experiments we need to participate in. They seem so tasty. So among the 8 sweeteners tested, isomaltulose seemed to be the most suitable for substituting sucrose. So therefore, in terms of both these factors, isomaltulose seems to be doing really well. Currently, a lot of health products available in the market have isomaltulose already. So, in even in things like yogurt, jam, juices, beer, a lot of sports supplement, even in child growing up formula and rehydrating solutions, and in meal replacements, you would find isomaltulose. So, I encourage all of you to read the food labels in some of the health supplements that are available over the counter to identify isomaltulose or see if you can find Palatinose written in these labels. Chang Chien was an undergraduate nutrition honors student at the International Medical University. For her final year research project, she looked at developing her own version of egg custard pudding using isomaltulose. Now let's zoom into her results and see what she found. Zooming into these tasty results, we see that she had 3 versions of her pudding and 3 versions of the caramel to serve it with. So here in this spider plot, you see the 3 versions that she used and how they were scoring in terms of sensory attributes. So what were the 3 versions? The first version was the original version with 100% sucrose, and this is shown in red. The second version was the 100% isomaltulose version, and this is shown in yellow. The third version was a 50-50, 50% isomaltulose, 50% sucrose version, and that's shown in green. So when you look at this spider web here, you see appearance, aroma, taste, texture, overall acceptability, and sweetness are the dimensions in which she has tested using these 40 subjects that she had enrolled in this sensory testing. So the only place where you see some difference is the sweetness. We know sucrose is 2 times as sweet as sucrose. But look at what happens when you're moving towards 5 in terms of sweetness. In terms of sweetness, moving towards 5 means it is too sweet. So whereas isomaltulose is around at the 3. 3 is just about right. So what this research showed us is there are particular speeds in which isomaltulose very successfully replaces sucrose. Thank you everyone for listening so patiently for so long. So now let's finish off with the take-home messages. Number one, with the evidence that was presented here today, it looks like isomaltulose is a very promising alternative to sucrose, especially in terms of glycemic management and body weight management. Number two, we understand mechanistically how isomaltulose works. Therefore, it seems to have potential for food innovation. More importantly, there is scope for long-term trials in the area of isomaltulose and its use for personalizing diets for health. Thank you everyone for your patient listening, and thank you again NSM and Benio. I would also like to acknowledge Professor Jordi Salas for his feedback on this presentation. And if any of you have questions or would like to discuss anything with me, please feel free to reach out to me on this email here or on the social media that's listed here. That's my faculty, and that's the really large research group that I'm a part of, and we'll be very happy to receive any of you. Thanks, everyone. Bye bye.